Assalamualaikum warahmatullahi wabarakatuh, and hello everyone. Let's introduce ourselves first. So my name is Afif Shafi'i bin Muhammad Uzi, number metric 205784.
My name is
Alia Najwa.
Okay, Alia, Najwa, you first.
Okay, my name is Alia Najwa binti Abdul Muttalib. My matrix number is 205220.
My name is Ahmad Daniel bin Zali and my matric number is 207138.
My name is Aino Hanani binti Rozmi and my matrix number is 205428.
Has everyone introduced themselves? Tonight we are going to discuss the question for practical 3, surface sterilization of least explant. There are 14 questions in total. So let's start with Alia Najwa. You can do question number 1 until 3. You can first.
Okay. While I share my screen, I will turn off my camera because I'm afraid that my line is lag. Okay.
Okay.
Can you see my screen?
Yeah.
Okay. So our experiment is about surface sterilization of leaf explants. Okay. So actually I have write my observation, but it in my tab it does not save. So I had to share screen this plain table. So I will talk about the result. Okay. So for sterilizing agent, we use 10% Clorox solution, but the difference is the exposure time, 5 minutes and 15 minutes. So for the 5 minutes, I will write. So for the 5 minutes, for the 5 minutes, you can see, right?
Not yet.
Okay, but never mind. I just talk lah senang. Okay, for the exposure time, 5 minutes, I prepared 5 expand. So out of 5, I assume 3 of them is contaminated. So the percentage of contaminated is 3/5 times 100. 100 equals to 60%. Okay, so when I assume 3 of them is contaminated, then the viable expand, I assume 2 of them. So it will make up 40% of the remain screen. Okay, so to know how much the dead expand, I relate to the number of contaminated expand. So just now I said that 3/5 is contaminated. So when the expand is contaminated, it will be dead and turns brown. So the percentage of the contaminated expand and the percentage of the dead expand is same, which is 60%. So how to know the plate is contaminated or not? So how to know that the plate is contaminated or not? I refer to the number of viable explant and the number of contaminated explant. So for the first table, we know that 40% of the viable explant is lower. So it's lower than contaminated explant, which is 60%. So I assume that the plate is contaminated because the number of contaminated explant is higher. Okay, so for the second table, for 15-minute exposure, I use three explant prepared, and from the three explant, I assume that 1/3 is one explant from the three explant prepared is contaminated, which is it will make up 33.33%. So the remaining two explant, I assume that it will remain green. So it will make up 66.66%. So how to know that the number of dead explant, how to know the percentage of dead explant, I refer to the number of contaminated explant. So when the explant is contaminated, it will become dead and turns brown. So the percentage of the explant contaminated again with dead explant is same, which is 33.33%. So how to know the plate is contaminated or not? I refer to the number of viable explant, which is remains green, and the number of contaminated explant. So the percentage of the viable explant is higher than the percentage of contaminated explant. So I assume that the plate is sterile, not contaminated. So can I proceed? Any question?
Anyone have question?
No.
No. Okay. So
Okay, I will proceed to the next page, which is the question. Okay, so number one, based on the observation, explain the result obtained. This is the one I explained earlier. Okay, so suggest a modification, if any, to improve the protocol. Okay, so the modification I suggest is that at the cleaning step of the procedure, we will include soap when rinsing the leaves so that any wax on the leaves or organic chemical on the leaves will be removed and it will not interrupt the action of sterilizing agent. Okay, so number two, based on the result, draw a suitable conclusion. So my conclusion, I can conclude that the efficiency of the sterilization is affected by the amount of exposure time of plant. So we can see that at the experiment number 2 with 15 minutes, for me, I think that is enough time for the explant to completely sterile. But when the time over the limit, which means that too long, it will also damage the explant structure, the explant. So my conclusion is the efficiency of the sterilization is affected by the amount of exposure time of explant with sterilizing agent. However, an excessive amount of exposure time of explant with 10% Clorox solution may result in dead explant, as they can handle the amount of chemical solution. So number 3, apart from exposure, explain other factors that may affect the efficacy of the sterilizing agent used in this practical. So I found two other factors about this. So first, type of leaf structure. Okay, some leaf surface contains wax that may prevent the sterilizing agent to penetrate the leaf. That is the first factors. The second one is physical-chemical environment. It will increase in pH. The increase in pH will remove the antimicrobial activity of the disinfectants while reducing the antimicrobial activity of others. The pH will affect efficacy by altering the disinfectant molecule on the cell surface. So that's from me. So any question?
No, everything is clear. Anyone else? Do you guys have question?
No, I don't have question.
Okay, before we proceed to the next video, I think I need to stop because we have reached our limit. So I'm going to post a new link and join again. Okay, let's continue with question number 4, 5, and 6. How about Akmal? Can you answer question number 4, 5, and 6?
Okay, so I will share my screen. Afif, can you make me as a host?
Okay, sure. Okay. Okay, share your screen now.
Okay, so you guys can see my screen?
Yes.
Okay, so for the question number 4, if the surfactant is not used, predict the outcome of the practical based on the percentage obtained above. A surfactant or a wetting agent such as twin helps wetting of the leaf surface in which it will reduce the surface tension significantly that allows the sterilizing agent to disperse more evenly and repel the air from surrounding. If the surfactant is not used, the percentage of contaminated explant will increase as the sterilizing agent did not have good surface contact and air or microbes could be trapped on the leaf's surface. So you guys understand the answer? You guys have any questions?
No question.
No question. So I will proceed to the next question.
Yes.
So for the question number 5, if the step that treated the explant with 70% alcohol was ignored, could the outcome of the practical be changed significantly and explained? So my answer is yes, because 70% of alcohol is a type of disinfectant that has an antimicrobial action mechanism by denaturation or dehydration proteins. If 70% of alcohol was ignored from the step, the outcome of the practical will be changed significantly, in which percentage of contaminated explant will increase because some microbes on the leaf surface might not be killed during sterilization. So you guys have any questions? No? So I will proceed to the question number 6. So it says that suggest a suitable sterilizing agent to replace Clorox used in this practical. Modify the protocol to suit the new sterilizing agent. So my answer is Clorox can be replaced with the calcium hypochlorite as the sterilizing agent because it is readily and available and can be diluted to proper concentrations. However, the concentration and time for the practical usage of calcium hypochlorite must be determined empirically for each type of the plant due to phototoxicity. So that's my part. So
Okay, that's clear. Anyone have question? I guess no. Okay. Can I ask Alia Najwa? Can you share your screen like just now? I want to explain the.
Okay.
Thank you.
Okay. Can you make me host?
All right.
Okay. This page?
The next page. The question. Okay, so I'd like to answer question number 7, 8, and 9. So for question number 7, name the type of sterilizing agent used in practical. So there are two types. There are Clorox and magnesium chloride, but our group will do Clorox. And for question number 8, name the active ingredient of Clorox and its concentration at 10% solution of Clorox. So the active ingredient of Clorox is called sodium hypochlorite. And the concentration of sodium hypochlorite at 10% of solution of Clorox is around 5 to 6%. So anyone have question for question number 7 and 8?
I want to ask, Clorox and sodium hypochlorite is the same thing or not?
Sodium hypochlorite is the active ingredient in the Clorox. So Clorox have a lot of chemical, but the active ingredient is the sodium hypochlorite.
Also, just now at the Akmal's part, suggest a suitable sterilizing agent to replace Clorox tu kan? Akmal suggest to replace with sodium hypochlorite. So, if replaced with sodium hypochlorite, hypochlorite is active agent
ingredient
in Clorox, right?
Yes.
Active ingredient. So that means we should suggest a new sterilizing agent lah kan? We should suggest another sterilizing agent lah kan?
It should be that. Do you have any suggestion? Or Akmal have anything to say? No. Alia, do you have any suggestion for other type of sterilizing agent?
Maybe mercury chloride.
Yes, mercury chloride can be used because the question asks because our group are doing Clorox, right? So you are suggesting we using mercury chloride. Is that is that what you mean?
Yeah.
Okay, so is that all?
Yes.
Yes. Okay. So can I proceed to question number 9?
Yeah, sure.
Okay. So question number 9. Explain.
Sorry. Do we need to just now Alia suggested the mercury chloride, right?
Yeah.
We need to explain because if number 6 we change the answer to mercury chloride, do we need to explain the modified protocol?
The modified protocol to suit the new sterilizing agent.
Because we stated a different answer, right? Just now. So do we need to explain?
I think we do need to explain it.
The protocol is different.
I think
I thought it's same. Oh, maybe different.
From what I read in the manual,
I think the difference is just the exposure time.
Just as the exposure time. So
From 5 and 15 minutes to 1 and 5 minutes. To use the mercury chloride. I think that's all.
That's all that is supposed to is.
But the procedure is the same, kan?
Just
only the exposure time.
Okay. Alright. Okay, thank you, Alia Farzana and Alia Najwa. Okay, so proceed to question number 9. So question number 9. So the procedure to prepare 10% of Clorox solution and mention whether you need to autoclave the solution or not. So the procedure how to prepare Clorox solution is first, make sure you are wearing protective gear while diluting the bleach, as the bleach can irritate your skin. So wear rubber glove and all the necessary things. And then you need to prepare the solution by pouring 1 part of liquid bleach and 9 part of water. And then it will yield 0.53% of sodium hypochlorite solution. Then, extra info, make sure to use cold water, as using hot water to dilute the solution will decompose the active ingredient of the bleach, making the solution ineffective. And make sure to store it in a plastic container for both mixing and storing, as if you mix and store it in a metal container, there is some reaction that can happen and make the sodium hypochlorite ineffective, same as if you use hot water. And after all that is done, store it out of sunlight, and that is all. That's all the procedure. Do we need to use autoclave? Cannot. You cannot use the autoclave for any solution that containing bleach, as when you autoclave a solution containing bleach, it will produce a toxic gas that will quickly corrode the autoclave, making the autoclave cannot work anymore, making the autoclave damage. Do you guys understand the procedure? And the reason why we cannot use autoclave? Everyone clear?
Yes, understand. Clear.
Clear.
So, all right, let's proceed to question number 10, 11, and 12. All right.
So I will stop sharing screen.
No, it's okay. I continue with the question that you. Okay, it's okay.
Okay. Right.
I'm sorry if I'm a bit lagging because my internet connection is unstable right now. Okay, I will continue with number 10. It said that justify the need to rinse and expose the explant with sterile water for a certain period of time after Clorox treatment. So the keyword here is what happened. What actually the need to rinse and expose to the explant after the Clorox treatment, right? Okay, so my answer is that it is to maintain the hygienic condition of the explant and to prevent their introduction of the contaminating microbes. The hygienic condition here is that the condition is free from the microbes. So basically, the need of the rinse and expose the explant to sterile water is to maintain that there is no microbes there. Okay, understood? Or anyone else have a different answer?
No.
Okay, I will continue with the next question. Okay, if rinsing with sterile water was performed quickly, without leaving explant in water for a while, what would be the consequence? Predict based on the percentage obtained above. So it state that predict based on the percentage obtained, right? So my answer is that the consequence based on the percentage obtained above is that the percentage of the number of the contaminated explant will be increased. Also, if the contaminated explant increase, so the percentage of the dry explants will also increase. So it is because the reintroduction of the contaminating microbes will always appear. It will be appear again. So it really so the consequence is it will increase the dead explants. Okay. Do you guys have any question? Okay, I will continue with the next question. Okay, number 12. If the source of explant was not heavily contaminated, which part of the sterilizing protocol could be modified to make it more effective? Explain. Okay, for me, I think that the sterilizing protocol that could be modified is the surface sterilization step, which is the exposure time when the immersed leaf in the 10% of the Clorox. Maybe the exposure time can be less than 15 minutes because it might damage the explant cells because it's that it is the explant is not heavily contaminated, right? So maybe there's too little of microbes, so it might be damage the explant cell. You guys have any different answer than me?
No.
Okay, I think that's all from me.
Okay, thank you.
Guna Alia punya je. Okay, so number 13, right? Number 13, the question is, if the source of explant was sterile, which part of the protocol could be modified? And it stated we have to explain. So my answer is, if the source of the explant was sterile, the explants can be prepared directly without sterilization. So it means that no part should be modified because this is to maintain the sterile conditions of the plant. Apart from that, most probably the use of additional disinfectant or sterilizing agent could damage the structure of the product of the plant. So anyone has any other answer?
No.
Okay, so number 14, suggest a method to grow a plant under sterile conditions for explant preparation. I would like to suggest micropropagation. Do I need to explain what is micropropagation? Explain.
Sure.
So I will explain like briefly, okay? Micropropagation is the process of multiplying plant stock plant material by micropropagation, the definition is the process of multiplying plant stock plant material by growing plantlets in tissue culture to produce a large number of progeny plants, then planting them out. So it means like micropropagation is like the production of whole plant through tissue culture from plant parts such as shoot and root tips, leaf tissues, anthers, nodes, meristems, and embryos. So in this process, plants can be propagated vegetatively by using small parts of living tissue of artificial growth mediums under sterile conditions. So it's like the things that we learned last week due to the totipontency of the plant. So that plant will like generate shoots and roots and consequently whole fertile plant under the sterile conditions. So also during this culturing process, the plant should be cultured in culture rooms where the environmental factors such as temperature and light are controlled. Do you guys understand?
Yes.
So
That's all.
that's it lah. Micropropagation. That's all.
Okay, so we all have discussed from question 1 until 14. And before we stop, do you guys have any question? Okay, so no, that's all. Thank you guys for coming tonight to discuss the 14 question. And Assalamualaikum.